hepatitis B virus and hepatitis C virus. These are the two important viruses which are very much common in Pakistan. Now let's look at the taxonomy of these viruses. Hepatitis B virus belongs to a genus Orthohepatna virus and the family is Hepatnaviridae, while hepatitis C virus belongs to genus Hepaciviridae and the family is Flaviviridae. Now hepatitis B virus or hepatitis C virus, they are naturally acquired during blood transfusion, which is very much common in Pakistan because of the improper screening of blood for donation. Apart from this, a vaccination is very important in order to control these type of viruses. So recombinant DNA vaccine is available for the treatment of hepatitis B virus. Now these viruses are transmitted among young generation by the process of tattooing, ear piercing, or by using inadequately sterilized instruments by the dentist specially. Now for the transmission of hepatitis B and C viruses, there are non-parental routes. They can be domestic content or it can be unprotected homosexual or heterosexual contact. Keep in mind that there is another type of virus known as hepatitis D virus. The important thing to mention over here is that if the individual is suffering from hepatitis B virus, then there are many chances that he or she will also suffer from hepatitis D virus, because a defective RNA virus requires the presence of hepatitis B virus for replication here. Now, if the co-infection occurs with hepatitis D virus, then hepatitis D virus will increase the or flare up the symptoms of hepatitis B virus as well. Now, if we look at the prevalence of hepatitis C virus as compared to hepatitis B virus, then the prevalence of hepatitis C virus is quite high in Pakistani population. And if we talk about the prevalence of hepatitis C virus antibodies, then it is slightly higher in medical care workers as compared to the general population. Now, hepatitis C virus commonly spread by a route known as parenteral route. Hepatitis B virus is present in blood products, urine, semen, CSF, as well as in saliva. There are chances of hepatitis B or C virus in lab personnel as it is transmitted by various ways. One is the parenteral inoculation, then the droplet exposure of mucous membrane, or it can be contact exposure of broken skin with these viruses. Now, always discard the sample after sterilizing it properly, because the virus remains stable in the dried blood sample as well for several days. Now, so far in the dried blood sample, we have not seen any avirulent or any attenuated viruses that has to be identified from the dried blood sample. For routine purposes, we identify hepatitis C virus in the blood or in the serum. Less frequently, it can be identified from the patient's saliva, लेकिन यूरिन और सेमेन का इस्तेमाल हम हेपेटाइटिस सी वायरस के सैंपल के तौर पे कम करते हैं. Now the sample appears unstable at room temperature, so they must be freeze at -20 degree centigrade. लेकिन अगर आप सीरम का इस्तेमाल करने लगे हैं, then thaw it before using it. लेकिन thaw करने के बाद उसको दोबारा freeze ना करें, because this will disturb the virus structure, integrity, as well as the survival of viruses. Now, if we are dealing with the hepatitis B and C viruses, then it's better to deal with such samples at Biosafety Lab Practices 3. लैब वर्कर्स के लिए ज़रूरी है कि वो हेपेटाइटिस बी के अगेंस्ट वैक्सीनेशन कराएं. नाउ लाइसेंस्ड रिकॉम्बिनेंट वैक्सीन इज़ अवेलेबल अगेंस्ट hepatitis B virus, but no vaccines are available against hepatitis C virus in humans.